Good morning, everyone. Please confirm whether my slides are visible to you, and am I audible? Please tell, am I audible? Okay. Now we are enter into the section. I welcome you all to this seventh day of CSIR online training. Last 6 days means last 5 days we are discussed about the cell communication, cell signaling, that kind of part. Continue to that topic. Now we are going to discuss about the topic basic concept of developmental biology. This is very important part in our CSIR examination because they are asking the question majorly from the three different subject, that is unit 4, unit 5 and unit 13. In this chapter they are asking a number of questions, so only we are start the section with these three topics. This concept developmental biology. What is developmental biology? It's this term is new to the biotechnology people, biochemistry people and microbiology people, but zoology and no, some other zoology oriented subjects peoples may know what is developmental biology. This is in this presentation I am going to explain with a basic concept in developmental biology. This developmental biology fully covered with the topic the human cell development, how the human cell develop in embryo, from embryo to the whole body tissues. That is the basic concept in this developmental biology. Small important terms are there. That terms are explained with potency, commitment, specification, induction, competency, determination, differentiation, morphogenesis. These kind of words are the basic words of developmental biology, that basic concepts. I'm in this presentation on 1 hour. I'm going to cover the two topics. As usual, I said this is not not covered by this 1 hour. I'm not able to cover the whole topic in 1 hour. I'm just give an introduction to you. Please refer the materials and you can get a better knowledge. Now I am entering into the topic. Okay, what is developmental biology? The small one line word. The pattern formation, the body tissue formation, or it's embryo to the human body development. This developmental biology combinationally explained with different stages. That stages are going to be part of it in the developmental biology. The process of embryogenesis involved in the coordinated cell fate control. What is cell fate control? What is cell fate? That cell fate, which means the cell having the specific fixed functional development. Okay, this cell can be developed as a epithelial cell. This cell can be developed as a or macrophage cell. That are the already fixed one. That are endangeredly fixed by the nature. That cells are not able to produce, not able to form some some other tissues, so that cells are not able to form to some other parts. That the cell fate control, that is the control. Okay, embryo can develop in various stage, develop to various cells. That's all in a cell fate control. This why I am stress this word means because upcoming slides I am repeatedly including this word, so only I am telling. Okay, next in the developmental biology, the pattern formation is a very important one. Okay, what is a pattern? If the pattern formation is major final step in the developmental biology, but the pattern formation is depends on the previous steps. That previous step means the embryo formation, development, embryo to tissues formation, tissues to organ formation, everything having the everything are all involved in the pattern formation. Okay, pattern formed because now yes, previously we discussed the cell communication, right? That cell communication is a major thing happen in the cell pattern formation. Due to that cell communication alone, that pattern is formed in the and it develop the organs. So that previous unit and this unit are partially interlinked. Okay, now I am going to discuss the important topics, important terms. The process of development. What is the process of development? In the process we have determination and differentiation. Okay, these two terms are very important. Determination. What is determination? This determination is, first we want to know the embryonic cell can be developed. It is possible to develop. It have the whether it have a potential or not. So the early embryonic cell is determined by the developmental potential is greater than the developmental fate. We want the rate is the developmental fate want to be higher than the developmental potential. Developmental potential want to greater than the developmental fate. And then after the embryonic cell development, that later period, the determined the developmental fate is fixed after the particular stage, that not able to determine. The determined cell is eventually differentiate after the this embryo is ready for the development. After a particular period, that cell next enter into the next level, that is the differentiation level, maturation level. Okay, so first level of embryogenesis, the first level of development process is determination. First we want to understand the potency of the embryonic cell. After it getting the potency, it enter that allow to develop, that is the differentiation. Differentiation means multiplication, and it's a group formation. Okay, another definition I am explained here because I'm refer many things in internet, but that period I am receiving some more informational slides, so I am included in this presentation. First, determination. When a cell is choose a particular fate, okay, we can't the embryonic cell that we are not all the cells can be produced the human human cells, human tissues. Some of them have alone having the potency. So first is the determination, that is the selection process. It's happen via the signaling process. In upcoming slides I will explain how what is the signaling process happen in the determination process. Determination is a receiving of the potent cell through the cell signaling. Okay, after determination, the cell can be differentiate via cell signaling. In this cell signaling process, different cell signaling process, in this step, that cell can differentiate the gene expression. So the gene expressing many protein, that proteins are make the signals and communicate with the other cells. That cells are produce that inducing the cell division. So in this after the determination, the induction happen in the genetic material. That induction is differentiates via the induction. Okay, induction is a very important term in the differentiation step. Okay, some biotechnology peoples may know what is induction. Once we are added the gene in a particular recombinant, we will do the induction on the cell. After the induction, that cell can produce particular specific protein that naturally happen in our human body. That is the induction in determination step and induction in differentiation step. Induction in determination step, that induction happen as a receptor protein. Receptor protein means that normal cells are maybe lack of the protein, so induction happens for producing the receptors on the cell surface, zygote cell surface. So the zygote cell surface having many receptor proteins, that proteins want to be in active signaling pathway. That signals are induced by the internal genetic gene expression. So the first become determined via induction. Determination also happen via induction. Another thing is a differentiation via induction. Okay, now this is the 5 thumb rule of the cell developmental biology. It's all things are explained with this 5 points alone. Okay, the first one is growth. Growth helps to increase the size. Okay, increase the size of the cell. Another one is cell division. Increase the cells, count of the cells. Third one is differentiation. It's a differentiation of cell type. Okay, that embryo want to develop into epithelial cell, endothelial cell, everything we want to develop, right? That is the differentiation. Diversification, that want to develop into diverse cell types. Okay, next, fourth one is pattern formation. That is key role playing step, that is organization. Another thing is morphogenesis. In this morphogenesis, the generation of shape and structure. Okay, after forming the pattern that want to develop in the particular shape and structure. Okay, after this steps happen happen to embryo, that embryo called as the adult. Okay, after this adult stage, that is called as the gametes. First is zygote. From that zygote, that embryo develop into all the stage. After reaching the particular 4 steps, that is enter into the adult stage. Then adult is called gamete stage. It's a 5 thumb rule of our okay. Next, first step I'm told, right? Differentiation happens. Differentiation happens in 2 ways. One way is diverge of developmental route. Another way is the individual development route. In why I am telling this in different way means because the different divergent cells development helps that way kind of is helps to form various tissues. Okay, but individual cell development it helps to the individual tissue cells, individual cells development. Okay, first image one cell having the diversely located constituents. These constituents are determined the further determined by the mutual interaction. Okay, this region interact with same cell, but this colored region interact with the same region, the white region interact with the white region, the blue region interact with the blue region. This kind of process happen in the further determination. After the determination step, primary determination, that ended to the final determination, that is the commitment. The commitment word is very important thing in the developmental biology. After the potential identification, the first step is a potency. Okay, red color want to interact with like that cells are formed as developing the specific character representing red color, and then green color represent a specific character, that cell having the specific properties that is able to produce into specific formations. Next, after the specific character development in the embryo, that want to enter into final determination. In this final determination, that cells are having the specific commitment, that cells want to group of the same similar cells, that same similar charactered cells are need to be develop the specific organs. These specific organs are so this diverge of developmental route, we have a 3 different steps: mutual interaction determination, the commitment determination, that is final determination, then the terminal differentiation. The individual cells are developed into different organs, tissues, that is the epitomic tissues, nerve cells and muscle cells, that kind of process. It's a differentiation of diversion of the compound. Why I am stressing the steps means they are given the steps in various form: specification, determination, final commitment, and the differentiation. These are the 4 steps in diversion of developmental route: specification, mutual interaction, that is the determination, then commitment, terminal differentiation. You want to take this 4 step into your brain because they are given is in order. You want to arrange this order for further. They give a mixedly they are provided in your options. From that options you need to find out the correct order. In this kind of question they are asking some direct and that asymmetric, symmetric way, that kind of way, ascending order, descending order, that kind of confusions are there also. So please understand the steps, that is want to specification, determination, commitment, differentiation. Okay, next enter into the individual cell development. Here the determination step, it's very important. Here there is compared to the region of development, individual development, with the individual development, that individual development don't have the determination step. That is one determination step. The mutual interaction is not there. Only one determination step, that called as a commitment. Specification, only one cell is committed to the cell division. That cell then third step is a differentiation. Specification, commitment, differentiation, that is the specification, only one determination method, and terminal differentiation. That after the terminal differentiation, that is formed the neuronal cells. These are the very important thing. This individual cell development happen with the unipotent stem cell. Very important term, very repeatedly asked question, that individual cell development happen with the unipotent cell. Unipotent cell means that is only specifically producing partics alone. Okay. I think it's a basic introduction. Move to the next slide. Okay. In this, after understanding the specification, that developmental process, first we want to understand what is commitment. Commitment is in a developmental biology means that cell is committed, okay, that want to produce some tissues. Okay, they having the 4 different stages. Naive stage, after the cytoplasmic determinant, that is called a specified stage. After particular period, that the loss of competence of alternative fates, that is a positive, okay, we that cells produced enough amount of tissues, okay, stop, that is a positive feedback loop. After that determined, that then cell specified gene expression happen in the particular cell, that is differentiated into various tissues. In this commitment, we have 3 different specifications, that is the autonomous specification, conditional specification, syncytial specification. Okay, what is specification? The point the cell developed autonomously, that is cell specific tissues. One cell can develop to specific tissues, that is called as the specification. Okay, this is please imagine this is a group of cells. It's a like embryo. In this from this embryo, specific cells are isolated. That isolated cells can be grown into specific tissues, that is called as the specification. In this conditional specification, what happened? For example, this is the plastula. In this plastula, it having the 3 different red color, blue color, and sandal color, biscuit color. That is 3 type of tissues are present in the blastula. We want to replacing the particular color specific blastula cell, normal black cells. So they are transplant to the back into the particular the belly region. So in this normal blastula, that red color that tissues are present at top of the animal. But the transplanted tissues, transplanted cells having the red color region tissues in the belly region. Okay, this is the conditional. Specifically, they are giving the condition. Okay, this cells need to be grown into particular this portion. This is conditional specification. And also autonomous, it's happen in all human body. The cell fate is determined fertilization, my fertilization by morphogenetic determinants in ovum. The morphogenic determinants, what is the morphogenic determinants? That is mRNA or protein that causes cellular commitment. That protein is having the signal, okay, this cell can be specified for the cell differentiation. This mosaic development, what another term is the mosaic development. Embryo function, that it like a mosaic of the independent cell differentiation part. Each and every cells are developed into the differentiating parts. the essential specification here. The essential formation means the cell-cell communication. Nucleus unconditionally multiplied in the cytoplasm. That morphogen may be the gradient within the cytoplasm. It having the specific quantity, that specific level. That's the essential specification happens when the cell interact with each other with specific signals. That similar cells are interact each other and form the tissues. That is the essential specification. Then this is the after the specification. Now previously we are discussed the specification. After this differentiation understanding, we are entered to the commitment, that is the specification. After the specification, that cells undergone the determination and differentiation. What is the differentiation and differentiation? Already we discussed about single in this differentiation. In the first step is determination. Only one region can develop into different region. That different cells are specifically differentiate into tissues. That is, determination is a random process, differentiation is a specific process. Here it's one simple comparison table. It's a very important concept understanding table. The cell differentiation occur at the totipotent and embryonic cell. One very important thing is the determination happen in the totipotent cells. Whether the cells are having the totipotent or not is determined by the determinant compounds. Determinant compounds are the cell signaling proteins. Okay, there in their experimental question, CSIR question, they are given the examples with the given the example and explain the same step. Okay, specification, determination, differentiation. These are all the three important terms happen in the developmental biology. So this basic concept is major role playing in the question understanding, because many of the questions are looks big, having a paragraph, but that answer is very simple. Just read the question with this basic concept. That imagine the questions with this basic concept. Okay, then that in this determination, the result of the asymmetric segregation of cytoplasmic determinant. Asymmetric segregation of cytoplasmic determinants. But differentiation is a gene expression. Okay. After the comparison, it's assign the fate of the cell. Okay, you want to do this. This cell want to do this. This cell want to do this. They are assigning. Okay, but the differentiation, the functional specialization. Okay, after the determination, okay, this cell can determine as for this capacity. In this differentiation process, that cell develop this specific activity. Okay, this cell want to develop into hot tissue. That is determined by cell determination step. After the determination, that specific cell differentiate into hot tissue. These are the steps happen in cell development, differentiation and determination and differentiation. After the embryo, determination happen. After the embryonic cell development as a with the specific characterization. After the specification, that is entered into the differentiation. Here happen the differentiation happen with different concept, that is the cellular differentiation. This cellular differentiation is explained with this image. First, the cell having the potency of a totipotent embryonic cell. That a totipotent embryonic cell means that can develop into various organs. That three are produced into endoterm, mesoterm, and ectoterm. These three type of cells are having the multipotent. Okay, this multipotent tissues are differentiate into various organs. This is the first step I want to tell, but this is a theory just about the cytoplasmic determination. This is a cytoplasmic determination because they understand this image after this zygote, that in the unfertilized egg, after allowing this cytoplasmic sperm nucleus to the egg nucleus, now that is called as the fertilized egg. Normally the unfertilized are having the nucleus. That nucleus is that cells, that cell zygote cell are allowing the nucleus of that sperm cell, that is called as the fertilized egg, that is zygote. After that after this stage, that is called that is enter into the mitotic cell division. That mitotic cell division, it's a two-celled embryo. It's a very important stage in cytoplasmic determinant. Cytoplasmic determinant alone developing the two-cell embryo development. So for the cytoplasmic determinant, we want mRNA, protein, and other signaling. This other signaling after this signal receiving from the external cells, that embryonic that zygote develop into two-cell embryo. Two-cell embryo happen with the cytoplasmic determinant. Then only development, that is differentiated into various organs. Okay, morphogen gradient. What is morphogen gradient? I think it is understandable. Okay, this morphogen gradient having the specific this is a intermediate step of determination and differentiation. After the cytoplasmic polarity, based on the cytoplasmic polarity, that cells are hung back protein gradient. Based on the hung back protein gradient, that are developed into various cells. So that cytoplasmic polarity, these are the various steps. There is the protein gradient formation, then gaps in gene, then is the pair rule gene. Pair rule gene, that similar cells are paired in the zygote in the zygote. Then fourth, the next step is the cellular blastoterm. When the cellular blastoterm, the segment polarity gene is forming the specific hemotic genes. Okay, it's a differential process. The multi potent having cell, that is a cytoplasmic polarity cell, observing the information cell signaling from the similar expressing cells, that cells formed into specific morphogen gradient. This, based on the gradient alone, the cells are differentiated into tissues and organs. Okay, this is the morphogen concentration based experiment. They are explained in an article. This information is very interesting and very understandable. Okay, first at the 0th time, that is no morphogen concentration, right? uh, Morphogen, that is a particular signaling compound. Then morphogen is after this cell is producing some low quantity morphogen in a low quantity. That is an embryo that after the receiving of signal from morphogen, the activated smart, that is a cell producing the smart compound, that is for gene production, gene expression. uh, so that is produced in the nuclear. The expression of gene is higher in the higher in the gene, higher in the particular cell. After the 2 hour it become high in the stage that that whole cells are occupied with the smart concentration. If the concentration of morphogen is high, that the gene expression of it's also high in the specific cell. The next concept is uh, if explained with the starting high with higher concentration. After the particular period, that gene A is over that is morphed with gene B, because gene B is overexpressed. After the particular fourth hour, there is no morphogens are there for signaling. uh, that is reaching the positive feedback. Okay, there is enough gene production happen in this cell, so please stop the addition of morphogen. That organ is formed, so please stop the the signaling. These are all the happen in morphogens are helps to express the protein for specific cell tissues production. This is another example with because various tissues, the skin, oral tissues are uh, crossing many stages: surface stage, tissue specification, genital stage, and differentiated cells. This differentiated cells form the various parts of body. uh, Here I am including this for just explaining what is the uh, role of the totipotent cell and pre-stem cell, and after the stem cell formation. Okay, the pre-stem cell program, linear establishments are happening in the cell. Here the cell linearage is a very important concept. In the linearage process, that cell is developed to different characterized cells. One is CDX2 character, another is FGF. This multipotent cell, okay, that is fixed. You need to produce this alone. This cell you need to develop into this cells alone. After this stem cell program lineage maintenance, that is they are identifying the multipotent cell. The multipotent cell is a TS that can be produced into any type of cell. Okay, next one is that differentiated cells are formed into pluripotent and multipotent. Here cells can differentiate into any organ. Pluripotent cells can develop into specific organs. Why I am said that pluripotent multipotent concept in this slide means, next we are going to discuss about the stem cells. Stem cells are a major thing in the developmental biology. You all know, I think, as a life science people, all of them understand, all of them having the knowledge about the stem cell. Here the one cell can be developed into blood cell, neurons, livers, everything. In the stem cell development, we have 4 different steps: totipotent cell, pluripotent cell, multipotent cell, nullipotent cell. Okay, what is the cell? How it is grouped into how it is classified. In the zygote stage, that is having the totipotent, capable of giving rise to all cell type of body, giving rise to all it can be developed into all type of this cells. Another thing is a pluripotent, it capable to rise all cell type of body. In this pluripotent cell, it is the determined cell. Okay, this cell want to develop this kind of cells, tissues, that is the fixed one. Another one is the multipotent cell, it can be developed into any tissue organ. Then nullipotent cells, not able to give any cell type. After reaching this to nullipotent stage, that cell not able to produce any tissues. It depends on the age of the embryo. In the embryonic stem cell, at the first 0 to 2 week, that is called as totipotent. Less than 100 cells are alone in the in the human cell system. Ovary, 0 to 2 weeks, there is only less than 100 cells are there in ovary. After reaching the 5 to 8th period, the embryo having the pluripotent. Okay, this can be grow into various cells. In that stage, more than 1,000 cells are there. And then 9 to 14th week, there is more than the 10 lakh cells are present in the developed in ovary. That cells are having the specific organal characters, that is a bone, that may be the that may be the skin. There was the developed partially developed human body is there. Then after reaching, after that is called as, after forming of all organs, that is called as the adult stem cell. I think it's understandable. It's an stem cell therapy, that is stem cell development. Embryonic stem cell, embryonic fetal and adult stem cells. These are all the four steps. Another I forgot to tell, this week's cell count, potency and embryonic stem cell name. These four are important for the CSIR examination, because they are given the cell count and mention the potency, asking like that kind of questions are there in CSIR examination, or it may be the match the following, it may be the paragraph like question. This is about the vaccine technology. Stem cell technology. In the stem cell technology, one cell, the outcome of this stem cell technology, that patient, the AD patients, the Alzheimer's disease, I think so, that patients want to get memory. They lost the memory, they need to develop the memory. Memory cells are not able to grow after particular period. So in this using of this stem cell technology, they are taken all these stem cell, that kind of stem cells that are developed into programming and develop into induce the pluripotent cell. After the induction of pluripotent, that is grown in the laboratory scale, that is on petri plate. After that, okay, this cell can be developed as a neuron, that form drug like structure, that injected into human brain. That person get the capability of developing the neuron system. It's a one way, that is an artificial way. Somatic cell, reprogramming, induction of pluripotent. After that, it enter into given as a drug. The another thing is naturally happen. Natural cells are, natural stem cells are the natural progenitor cells. Natural progenitor cells forming the neurons and glial cells. That cells are replacement therapy, and using this replacement therapy that they are injected the developed cell into their brain. Then genomic equivalence. After this are all the now we are entered into the stem cells, right? Stem cells can capability of development organ. In this genomic equivalence, they are having the instruction. Okay, after receiving the acceptance, that want to develop into various organs. That various organs are explained based on the nucleus capability. Okay, genomic in this genomic equivalence, that every cells are having the are taking equal amount of the donor nucleons. It's various models were used to explain how the cell division lead to differentiation. It's a nuclear determinant are segregated by the cell division, and resulting is a unequal nuclei. It's happen. So in this, it's a representation image of genomic equivalence, that is want to be in equivalent, otherwise the cells are misleaded. Next to the genome equivalence, okay, we are receiving the correct quantity of genetic information, correct quantity of cells. Then imprinting. What is imprinting? That is similar to the the first generation, the mother cells having the similar daughter cells. In this genome imprinting, we have two different types. One is a maternal and another is parental. Maternal imprinting is the that both AA and capital AA and capital small AA. They are capital A is male, and another one is a female. That for next generation is the first generation. Next generation it's a progeny only, expressed as a dad's the male allele. They are asking many questions about this topic, so please listen. Genome imprinting having two different type. One is a maternal imprinting, another one is a parental imprinting. In this maternal imprinting, that cell having that first generation, that mother cells are having the major father dad character, because dad is a black one. So this next generation, that is daughter cells, are having both progeny only, that having only the dad character. Then parental imprinting, in this expressed on the both are having the similar activity, but size of that is vary. That based on the first one is the color based allele, another one is the size based allele. That one is in the second generation, the daughter having the similar character of the mother, because the height is also similar to mother, but the male having the similar color of the mother and similar size of the father. So that male having both character of the mother. First, in this parental imprinting, the male having the color of the mother and size of the father. So it's called as the parental imprinting. I think it's understandable. Parental imprinting, just understand the concept. Based on the concept alone, they are given as a paragraph question. So please understand. Once I will repeat. Maternal imprinting is the character based one, that character that any one of the parent character, that may be the dad character, that may be the mother character. In this first maternal imprinting, they are expressed only the mother character, father character, sorry. In this first image, maternal imprinting, the second generation, that is the mother and daughter and son having the similar character to the father. There is a suppression of the particular mother character, so it's called as the maternal. Another one is the parental imprinting. This in this image, second image, second generation having the girl having the same color and image size of mother, but this man, the son, having the color of mother and size of the father, so it is a parental imprinting, that having the both mother and father character, so it's called as the parental imprinting. Okay, it's another one example with the laboratory technique, laboratory experiment. What is the mutated? How can we explain it's a mutated mother? Determine the mutated mouse, mutated gene. It's a normal size mouse, wild type. It's a small one. It's a mutant. In this mutant cell, they are having the expression of IGF2 in the normal cell, but in the after the mutation, this is not happen and not expressed in the particular cell, but that gene is in the normal cell. IGL is not also not expressed, because why here the color change they are indicating means, in a normal cell there is the IGF allele, that is the normal allele, that is for responsible for this shape, size, but in the mutant cell there is the replaced one. Okay, the next step of the mutation, that cells are lacking of the mutant expression, so normal gene is expressed in the mutants, expressed in the mutation determination. This is the 4 example. It's a mother one, the father one. If the next to this step after the father and mother gene expression, that is enter into the developmental stage. Okay, the first is imprinting erased in the here that may you may erase this character or may this erase this character, happen in the first step. Enter into the parental imprinting. Okay, that after the erasing, that gene may have the parental character, the mother character, or may have the father character. That for after reaching this it may be the parental imprinting, or it may be the maternal imprinting. After reaching this, the sperm and zygote is enter in, the oocytes enter into the sperm enter into the oocytes that form the zygote. After the zygote formation, that blastocells are developed. After the blastocell development, the embryo will form. It's another one example for the mutant and determination. Here the mutant determination questions are majorly asked questions in CSIR questions. Please understand, and I will send some materials for mutant and mutant related part. Understand the questions. That mutant alleles are imprinted. Here there is here it's maternal, like look like mother, it's look like the father. So mutant allele imprinted, no phenotype. No, there is no phenotype. It's a mutant. Here this specific mutant male. In a normal, there's a combinational one. The normal one is active on one allele is imprint. Here there's happen some imprint, right? One is imprinted. Here the mother is muted, so normal allele imprinted mutant phenotype. Because mother character is a major one, because that is the oocyte character. Uh, so here that mutant, if there is a mutant female, normal allele imprint happen in the phenotype. Mutated female involved in the zygote formation, the normal allele imprinted mutant is a phenotype. Okay, after this mutation happen, because it mutations, stem cell formations are happen in the differentiation or determination step. It want to be developed into organs. That may be the that after the development we can engineering the cells after understanding, right? Each and every step you are all understand it in this till this slide. Okay, till this slide you are all understand what happened in the human cell. So only here they are engineering the zygote, engineering the gene. They are modifying at the earlier stage, so that is called as transgenesis. This transgenesis process, we have the different terms, that is a chimeric animal, knockout, mutation. What is a chimeric animal? That can be carry an altered gene introduced using the manipulated embryonic stem. That it can be carry an altered gene, it having the altered gene introduced into the manipulated embryonic stem cells. Then another one term is knockout mutation. In this knockout mutation, there is a replacement of cell. Okay, it's happened by the homologous recombination. Here after the mutation, transgenesis means it may be a mutation, it may be alternation, it may anything happen in the transgenesis. How can we determine this? There are some examples are there. Here first one is a parental one. Here FLP is a recombination material. After the recombination you can see the homologous mutation happen. Okay, same site, same place, mutation happen. So that is called as a homologous mutant. Homologous wild type means it having both character, this wild type character. Another one is the heterologous, one gene altered at one particular place, another gene is altered at the another place. These are all the heterologous. Here an alteration happen in one place, next strand alteration happens in another stage. The homologous and heterologous mutation is another important term and conditions have involved in the developmental biology. It's just another examination. These are all the exam type questions, how they are involved. Okay, the production of gametes. Now it's full of the explanation about the production of gametes. After first we understand the what happened in the cell development. Now we are enter into the production of gametes. First step is a mitotic division. Second step is a meiotic division. In this way, steps are happened for cell division. Okay, these are all the steps involved in the sperm entering into the oocytes. This cell surface receptor based signaling. This cell surface, oocyte cell surface, having some receptor to receiving the signals from the sperm cells. Sperm cells makes the contact with the egg by the receptor binding. That is the acrosome. That is called as the acrosome. Then a second step is the acrosome react with the sonopellucida, that makes some release of the cytoplasmic particle. After the release of the cytoplasmic particle, that particle damaging the cell surface of oocytes, that is a hydrostatic enzyme. That hydrostatic enzymes are interact with the cell surface receptor and make the interaction with the oocyte. Then third step is agrosome react with the perivitine space, that is the intracellular space, that having many protein receptor. That protein receptor interact with the surface receptors of that sperm cells. Then entering of plasma membrane of sperm and egg fuse. Now that after reaching the intracellular region, that are removed from the external cellular region. That external cellular regions are degraded by the proteins present in the intracellular of oocytes. The sperm nucleus enter into the oocyte eggs. After this oocyte eggs are then final step, the cortical granules are fused with the egg plasma membrane, which membrane render vertellin layer. Okay, this after this step, the sperm enter into the nucleus. These are all the sequential steps happen in the fertilization. It's another representation. Okay, next is the embryo development. Embryo development is having the different step. First is the endosperm, one sperm fertilization center, and initiate the endosperm development. After that, that fertilized egg formation, it's produce zygote. Then the cell division happen. First is endosperm initiation, fertilization, zygote formation, and embryonic cell formation. These are all the happen in the embryonic cells. This is the step-by-step process. First, wall of ovary entry, nucleus present in the ovary, that allowing the entry of sperm cells into the nucleus. After entering of sperm nucleus to oocytes, that forms the that allowing the meiosis process. So 4 haploid cell formation, it's a very important step. After the meiosis process happen in the human cell, that is developed into 4 haploid cells. That 4 haploid cells are forming the embryo sac. The 3 cell generation cell. First one is a 2 cell generation. Then another is a 3 cell degeneration. That degeneration is happen with the embryo. Mitosis happen with the 3 types. That 3 types, after the 3 types, the embryo sac with the 8 haploid nucleus, that having the 2 polar nucleus. These 2 polar nucleuses are having the this formula. If one polar nuclei present in the cell, that is developed into a egg. Each and every polar nucleus are developed into egg cells. The only one egg cell is finally enter into the fertilization process. These are all the sequential steps. First one is the meiosis, 4 haploid cell formation, 3 cell degeneration. First is the 2 cell generation, another is a 3 cell degeneration. So one embryo formed in the sac, one embryo sac is formed. After that, 3 times mitosis happen in the happen to the cell. After this, that from the embryonic sac with 8 haploid nucleus, that 8 haploid nucleus having two polar nuclei, that two polar nuclei showing the forming the egg cell. Another one concept, the double fertilization. Double fertilization happen in the plant. This is about the double fertilization happen in the plant. Here first we want to understand the parts of the fertilization parts of the plants, the antipodals, polar nuclei, and egg cells, synergicytes. First thing is a release of sperm cells here into the oocytes. Yes, that, and after the entry, there was a two nucleus are present on the oocyte nucleus, that forming the two zygote. Two nucleus are there in the entered in the oocyte nucleus, so there is the two zygotes are formed. So double fertilization happen in the plant. Then zygote formation. In this zygote formation, different stages are there. This corona is not disease. This is a structure. It's a like movement. Movement is called as the corona radiata, zona pellucida. Then penetration, then male pronucleus formation. It's a metaphase plates. It's an day by day improvement of cells. Day 1 is a 1 cell 2 cell formation. Day 2 4 cells. Day 3 the 8 cell formation. Day 4 is a morula formation. Day 5 is the blastocyte formation. After that is the after this step alone, it's implementation. Okay, day 1, day 2, so after the 5th day alone, it implement at the in the ovarial path. Then after its cleavage, it's called as the rapid cell division. This rapid cell division happened after the zygote formation. The cleavage happened in every step. Here that 8 cell stage, after the 8 cell stage, is rapidly cell divided into the blastula. That blastula having the internal space gastrulation. This gastrulation is a very important thing in the cleavage, and gastrulations are both are most important term in the cell development. Here after the 8 cell stage, that divided rapidly form the blastula. After the blastula, in the blastula forming like one structure, that structure having the gastrulation region, that is called as gastrula. Okay, here we having this blastula formation in the different stage. It's another example, previously same example. Why that 1 minute. Okay, these are all about the cleavage experiment. Here this in this cell development, first is the somatis development, gilly cell development, free limb development. That are the development of the cells. Here some of the important terms are there in the cell development. It's a most important thing. I wish to take 5 more minutes. Please be patient, because this is the very important topic about the cell development. So only I am give the more important. First thing is the ectoderm. Ectoderm is the outer cell surface of the blastula. Outer cell surface. Another is the endoderm. Ectoderm is the outer cell surface. Ectoderm is the internal path. It's a cerebrous forebrain induction. Then outer membrane having the anterior, is called as anterior, that is a head specific. Then activation, transformation period. Here we have the posterior. The posterior is the retinoic acid, that having the retinoic acid. In the another part is the dorsal mesoterm. Ectoterm, endoterm, dorsal mesoterm. Mesoterm is present in the blastocytes. There another is called as a gastrulation. Already I am told, right? Gastrulation is the process that establishing the 3 germ layers. Because of this, after finishing this step, that is it's a full blastula, right? That want to be developed into various organs. So that this gastrulation process forming 3 steps, 3 parts: endoterm, ectoterm, mesoterm. Endoterm is the inner part, ectoterm is the outer part. In between them we have the mesoterm. Mesoterm is a development to this only development into organ. The ectoterm forms the exoskeleton. Important thing, ectoterm is forming the external skeleton. Mesoterm forming the organs. Endoterm is forming the inner lining organs. Inner lining means specific inner lining organs. The layer 2 layers. Okay, these are the important things. Here I am going blastula, the gastrula formulation. Another is the gastrula is gastrulation forming the various 3 differentiation: the ectoterm, endoterm, and the mesoterm. External ectoterm forming the skeleton, outer skeleton. Endoterm forming the inner line of organs. Mesoterm forming into organs, various organs. These are the 3 germ lines. These are the different cells are forming in the various lines. This ectoterm formed as epidermal skins, epithelial lines, mouth, nerve systems. Mesoterms forming the skeletons, muscles, skin, internal parts, internal organs, epithelial cells. Endoterm inner layer, that cells are forming the respiratory tract. That is a very urinary bladder. The endoterm is the inner layer. It's a very important region, because it can produce the major organs. Another example, pictorial examination. In this exam, it's a very development of the embryonic cells. This is the stage by stage. Now it's another thing happen. What is the symmetric in the plant? How the establishment of symmetrics in the plant. Plants leaves are look like similar shape, right? How they developed. First is the lateral mediator, and this primordium. In this primordium they are developing the leaf. That leaf is that is the first stage is the lateral, the second stage is the primordium, third step is the developmental of leaves in the then that developed leaves or petioles. That petioles are having the distal structure. This is the establishment of symmetrics in plant. We have some more example. I will explain in the I will give a notes. These are the first step by step process. First step is a founder cell requirement. Okay, this cell want to get us a first step. 3 axon establishment. Okay, we want to develop into 3 different shapes, that 1 step, 2 step, 3 step. Okay, first is 3 axis establishment, that the blade initiation. After the blade is initiation, we have the petiole and blade. After blade, first blade expansion is here. Okay, here in this portion that cells are allotted. Okay, we want to develop. Next step, the next blade, first blade, second blade, third blade, right? These are the steps. Then seed formation, this is the final step, seed formation and seeds germination. The seed formation is a basic thing. If you have zygote, here we have a 3n of endosperm nucleus. We need the basic seed form. We need 2n zygote and 3n endosperm. The zygote grows rapidly in a mitosis to form the embryo. Embryo consists of the plumule. Plumule is the future shoot formation and the root formation. These two very important in the seed formation, shoot formation and the root formation. Another one is the food stores needs to germination. We need the source foods, right? That cotyledons, that are called as the cotyledons. The plumales are shoots, radial are root, the cotyledon stores. Remember this, plumales are the shoot, radial are root, other cotyledons are the storages. This formations having the 2 different stage, endosperm seed and non-endospermous piece. These are the 2 differentiations between the endospermous seeds germination and non-endospermous seed germination. In this, having the presence of present and absence of endosperm, we having the different type. In this type, food is not stored in the but the non-endospermic, the food is stored in the cotyledon. The cotyledons are within the thin layer and properly arranged, but thick layers, and here the endospermic seeds are thick and freshly formed. In the endospermic seed formulation, the perisperm does not develop from nucleus, but the non-endospermic seed, that perisperm nucleus. Very important thing. It's a basic concept, but you want to remember. Okay, examples for the non-endospermic seeds, pie and then non-endospermic seeds is maize and castor. Okay, seeds germination. It's another final example for seeds germination. In this seeds germination, we have different example. First one is a garden bean. Garden bean is a hypocotyle. That is that having the radical. After development of that, cotyledons are developed and foliage leaves are formed. After the foliage leaf, the epicotyle and hypocotyle are developed from the cotyledon. From that cotyledon stage, the 2 difference are formed, that is the epicotyle and hypocotyle. In the corn, the coleorhiza is seed that having different, the coleoptile and radical. These 2 are developed in the foliage leaf. Then the pie is having the radial and hypocotyle. First here coleoptile, here it's called as the hypocotyle. They have 2 different regions, the foliage leaf and epicotyle. That epicotyle is developed from the cotyledon. These are all the steps, various steps involved in the seed germination. Why I am mentioned these 3 means the garden bean and the corn concept are repeatedly asked in the CSIR examination. The concept is very important. That's coleoriza and radial that developed into coleoptile and radical is developed into developing the foliage leaves and the coleoptile. Here next one is in the garden bean. The hypocotyl is developed in, developing the radical. After this step, the cotyledon and developed from the cotyledon, the foliage leaf, epicotyl and hypocotyls are produced. These are the final step, the seed germination. This now we are discussed how the cell developed in the human and as mammalian cell. How the cell developed in cell developed in the plant. It's an initial stage. The initial stage only we have discussed the seed formation, seed germination. Okay. Next tomorrow we will going to discuss the morphogenesis. How the cells are forming the outlook. This is all about the morphogenesis. Tomorrow we will discuss about these topics. Thank you for your support and cooperation. If you have any questions, please ask in the comment box. What is the difference between multipotent and nullipotent? Multipotent cells having the ability to develop into any organs. Nullipotent cells not able to develop. That are fixed one. That is IGF means interleukin like growth factors. That growth factors are useful for cell development. Thank you everyone. Thank you so much for your patience. Tomorrow we will see at 6 a.m. Thank you.